welcome to our annual Frontier Night. I'm Susan L., Vice President and Executive Director of the Foundation for Barnes-Jewish Hospital. We're so happy that you could join us for this inspiring and educational evening with some of the brightest minds in medicine and research. Tonight, we will have an exclusive behind-the-scenes look at the science fueling new discoveries and hope in the world of cancer. The life-saving work at Siteman Cancer Center is possible because of your generous contributions and giving at the Foundation's annual Illumination Gala. Let's hear it, it's coming up, right? That supports the Cancer Frontier Fund. The Cancer Frontier Fund is designed to accelerate the pace of discovery so that new treatments more faster from the lab to the patient. This year, we're excited to celebrate 15 years since Bill and Amy Komen established the Cancer Frontier Fund at the Foundation. As a two-time cancer survivor and passionate leader, Bill recognized the vital importance of cancer research and was determined to change the face of cancer. And what a legacy he and Amy have created with the Cancer Frontier Fund. We're forever grateful to the Komens, our good friends, for their vision and generosity. The Komens continue to galvanize the community behind cancer research. Donor investment in the Cancer Frontier Fund has launched more than 162 research projects at Siteman to fast-track breakthroughs targeted at nearly every kind of cancer. These research projects have evolved into groundbreaking new approaches that includes breast cancer and melanoma vaccines, better treatments for pancreatic cancer, breakthroughs in leukemia and lymphoma, and so much more. Your generous gifts at the Foundation's Illumination Gala and other ways make this pioneering cancer research at Siteman possible. Illumination's success depends on your generosity and the wonderful dedicated leaders who step forward to make a difference in our community. This year, we're honored to welcome Carolyn Kindle and Julie and Rusty Keeley as our lead ambassadors for Illumination. Carolyn is the CEO of St. Louis City SC, the first majority female-owned club in MLS history. She brings her entrepreneurial spirit and fearless leadership skills to build a winning team of Illumination ambassadors and supporters. Julie and Rusty have long-standing commitment to cancer research and have been active with Pedal the Cause, our community partner in cancer research fundraising. Rusty is the CEO of the Keeley Companies, a St. Louis-based company committed to giving back to the community. Both personally and professionally, Julie and Rusty are well known for their generous hearts and caring spirits. We're grateful they're sharing their leadership, talents, and passion for serving others to spark more support for cancer research this year. So now, let's please welcome Rusty Keeley. Rusty Keeley.
On behalf of Julie and Carolyn, we are truly honored to partner with the Foundation for Barnes-Jewish Hospital to the power wheel of innovative research of Siteman. This is such an important effort that impacts our friends, family, community, and people all around the world. And it starts right here in St. Louis. My personal philosophy at Keeley is if you get the people right, you get the results. And that's why we're here tonight, because we have the right people. We're together, we can achieve phenomenal results in raising more support for cancer research to make a difference. The brilliant physicians, scientists at Siteman are also the right people to get results. And they're getting results in the form of breakthrough treatments that save more and more lives. Siteman is a leader in treatment, research, prevention, education, and community outreach with nearly 600 Washington University physicians and scientists focused on the latest in cancer treatment and research. Your support of the work is critical as we continue to build the momentum in our fight against cancer. As the amazing Dr. Eberlein says, we want to make cancer a chronic disease, and we're on that path. Some of you have fought the cancer battle and are here with us tonight, or you have loved someone's hand, held it through treatment and worry. You understand how important cancer research is to finding better treatments that are more effective with fewer side effects. Thankfully, Siteman physician leaders and scientists are laser focused on this effort and making incredible strides. What you will learn tonight will spark your imagination, inspire your hope for a healthier future, and fuel your passion for making a difference in our collective fight against cancer. No one fights alone, and we're in this together. You're here because you're committed to cancer research, just like we are. Carolyn, Julie, and I are honored to link arms with you as we work together to raise record-breaking funds to continue to fuel cancer research at Siteman through the Illumination event. Before we dive into the evening, I want to introduce to you the amazing Dr. John Lynch, president of Barnes Jewish Hospital and president of the foundation as well. Dr. Lynch provides a critical leadership link between the Foundation for Barnes Jewish Hospital, Siteman Cancer Center, Washington University School of Medicine, the hospital, the physician scientists, and the many, many extraordinary team members who care for patients. Please join me in welcoming Dr. Lynch.
Well, thank you, Rusty. We're grateful to you and Carolyn and all the Illumination ambassadors for rallying support for the Foundation's Cancer Frontier Fund. Our ambassadors are vital to engaging more people and sharing the excitement about Illumination and the Cancer Frontier Fund. You are our secret sauce. You're our superpower. You challenge us, you represent us, you support us, and you connect us to our community. Many of our Illumination ambassadors are here with us tonight, in addition to Carolyn Kendall and Julian Rusty Keeley. Our 2024 ambassadors are Nancy April Stubblefield, Laura Ellenhorn, Stephen and Erica Harris, Tom and Jennifer Hillman, Linda and Jesse Hunter, Ashley and John Kemper, Joe and Kim Koenig, Bill and Amy Coleman, Jim Meyer, Susie Sachs, Dalton Schumacher, Kathy and Dave Tacklet, Ryan Todd, Lee and Lori Walensky, and Chris Zimmerman. Thanks to all of you for your dedication, your time, and leadership. Will each of you stand up so we can represent you? We can thank you if you're here. Yeah, and those ambassadors, Susie, I know Nancy's here. Thank you, Rusty, of course. As you know, cancer is an extremely complex disease. However, our researchers are making progress every day, but we have much more to do, and that's why we need you. Tonight, Dr. Tim Eberlein, the director of the Siteman Cancer Center, will moderate our panel discussion featuring some of the leading physician scientists in the country who call Siteman their home. Dr. Eberlein has led Siteman since its inception 25 years ago, and he finally gave me a pin tonight. Thank you, Tim, after 25 years. And has been instrumental in creating one of the 5 largest cancer centers in the country, and quite honestly, it's the best cancer center. The fact that Siteman Cancer cares for 75,000 people each year, including 12,000 who are newly diagnosed, clearly demonstrates the reason why we're here tonight. These patients and families are counting on us. So thank you for being part of our important mission to fund more life-saving research through the Cancer Frontier Fund. Now, Tim, would you come up and introduce our panel? Thank you so much.
Alrighty, thank you very, very much. John, that pin looks really great on you, especially for a pulmonologist. I mean, anyway, I wanted to just begin by making a couple of points. First, I wanted to thank all of you for being here tonight, because as Rusty and John said, you are the secret sauce. Now, how does this all work? How's the sausage made? Well, you give us unrestricted dollars. And it's like it were a venture capital firm. We, and Sheila's on our senior leadership, she deals with this every 6 months. We carefully vet each idea, peer review it, vet it again, and then decide which projects will become the innovation, future, new treatments, new paradigm shifts in the care of cancer. And when that happens, what do these investigators, and you're going to hear from 3 tonight, what happens? They write grants to the National Cancer Institute, other NIH institutes, and we are able to get more research grants, which accelerates the pace of discovery. In the past 5 years, we've had a 122% increase in these multi-investigator grants. We're really good at putting teams together. And all of you who are in industry and business, you recognize that's the key to really having a well-functioning business. You got to have teams that work together, have great leadership, and you're going to hear from a couple of examples of that tonight. So I want to thank you, because without your steadfast dedication over these last 15 years, no way we would be where we are. Literally, from a year ago, we had a 25% increase in NCI funding. And we're now one of the 3 or 4 top funded NCI centers in the United States. So that's all come about because of what you have done. So thank you. So that's point number 1. Point number 2 is we never rest on our laurels. And so we're always challenging the status quo. Some of you, I think, have seen the very large building on Forest Park Parkway, and that's a new ambulatory cancer building. And we are doubling down on the efficiency and patient-centeredness of our care. How is that? Well, we're going to cohabitate specialties. We're going to reduce the number of visits that people have to make, and we'll provide the experts to the patients rather than having them go hither and yon around the campus. Radiology is in that building, and there's nothing else but cancer in the building. So the vast majority of patients will probably go to 1 floor. Some may have to go to 2 if they get x-rays and special lab tests, etc. But again, it's an opportunity for us to really double down our effort, which has made Siteman Cancer Center, frankly, very unique. The care that we provide, the hope that we provide. And again, wouldn't be possible without the partnership between Washington University and BJC. Last, I want to tell you what a pleasure it is for us to have the talent that we have at Siteman Cancer Center. I'm actually amazed. Every day I go into the office, I get to witness a miracle. And I witness a miracle not because there are miracles to be had. It's because of the innovative minds, the passion, the work that goes in to creating these new projects and new paradigm shifting treatments. And so I say I have the best job in the world. Thank you very much. But it's really people like I'm going to be introducing tonight. They're the ones who really make everything possible at Siteman Cancer Center. And our patients are the beneficiaries. So, without further ado, I want to introduce our first speaker tonight, Sid Perham. And Sid actually came here with a vision of, can we develop a center here of excellence to really elevate the treatment of head and neck cancers. Now, Siteman, as I mentioned, has already put together multiple centers, leukemia and the blood cancers, in pancreas and HPV cancers, in brain cancers and other areas. And so it's great to have Sid's passion and his leadership. Without that, we would never be able to have a world-class head and neck cancer program. So, Sid?
All right, it's really my pleasure to be here, and let me lead by just emphasizing what's already been said. We are just so grateful for everyone in this room. I hope today I can show you a very small snippet of what we're doing with head and neck cancer at Siteman and give you a flavor of what the future looks like, give you something to dream about, envision, even talk about with your friends that maybe will affect them or maybe at least gives hope to those that we're still trying to better their lives. So I want to really focus on kind of frontiers that exist in head and neck cancer. And I have to lead by just saying, many people don't realize, but head and neck cancer is an incredibly common cancer. It's actually the 6th leading cause of cancer in the entire world. And these cancers typically arise from the lining of the mouth. I like to tell patients that as a head and neck cancer surgeon, I treat everything above the collarbone except for the spine, the brain, and the eyes, which narrows it down to basically everything else. So that's what we do, and that's what we treat. For years, people have thought of head and neck cancer as a disease of smokers and drinkers, and frankly, it was. And I think that's a big reason why the treatment for head and neck cancer has long lagged behind the treatment of other cancers. There was a certain degree of stigma, certain degree of blame on patients for causing their own cancer. While I certainly don't agree with that attitude, I think it is a reason that drove a lot of where we are today. But what's actually changed the face of the way we view this disease is the HPV epidemic, human papillomavirus. So if you look at college students, 80% of college students have HPV. By the time you look at 40-year-olds, that number is down to 20 or 30%. So the reality is this is a virus that's floating around and basically everyone is exposed to it, and a large number of people clear it. The problem is if you don't clear it, you're at certain risk of developing a cancer. And the rapid rise of these cancers, as you can see in that light blue line, has led to a massive change in how we look at head and neck cancer. These are cancers that come up in people that do not smoke and drink. And as of 2024, the head and neck cancer is more likely to be caused by this virus than by smoking or drinking. Just changed this year. So at WashU and Barnes, we're very lucky to have an incredibly strong legacy to build on. This is a picture of Joe Agura, who was the 5th chair of otolaryngology head and neck surgery. And Joe was famous for walking around the clinic with a pipe that he used to smoke for years and years. It was a different time. And actually, a funny story about the pipe, he actually ultimately was found to have a little spot in his lung on a chest x-ray. So he ditched the tobacco and kept the pipe for the next 20 years. And Joe was really a luminary. He actually completely invented the field of head and neck surgery. And at the risk of offending any of my general surgery colleagues, up until that point, head and neck surgery was largely living within the general surgery domain. And Joe sort of wrestled some of that away to say, we really need dedicated surgeons focused on this specialty. And so just to give you a perspective of how far we've come, in Joe's day, if a patient had a cancer of the throat, an area we call the oropharynx, to get to that tumor, they would split the lip, they would split the jaw, they would open up the jaw like a book, and then get to the tumor. It doesn't take a physician or a scientist to realize that that was a pretty tough thing for patients to get through. And they were in the hospital for weeks. Many never swallowed again. Many left with a tracheostomy. Today, we can do this surgery using a robot that all works through the mouth. A surgery that used to take 12 hours takes us 3. And we can use these robotic arms with a surgeon that controls those arms from a nearby console and a resident who assists at the head to do everything through the mouth with no cuts in the mouth to remove the tumor. And then we separately go to the neck to remove the lymph nodes. These patients leave the very next day sometimes. So this is a picture of the robotic arms coming in. We put a suspension into the mouth, kind of like a sword swallower at a carnival, suspending the patient so that we can see in the areas of concern. So we have all these great technologies, great research, great staff, a legacy, but what are we missing? We're really missing integration. We want every patient to have access to the amazing care at Siteman and at Barnes. We want to engage in high-risk, high-reward research. Although Tim mentioned we have wonderful access to NIH resources, the reality is it's exactly the people in this room that accelerate things that NIH doesn't fund. NIH doesn't want to take a gamble on something that has a 5% chance of working. But the thing that has a 5% chance of working is exactly what will cure these horrible diseases. And so we want to translate things better from the bench to the bedside and develop coordinated efforts. And it's these things that really motivated us to establish the first head and neck tumor center in the country. Up until this point, there were a few head and neck cancer centers, but that really didn't mean much except for maybe a navigator who holds your hand through treatment. This is so much more. Thanks to Dr. Lynch and Dr. Eberlein, we've been able to get this off the ground from its conception to its establishment in just 6 months. And from that point in last September when we established it, we already have 5 or 6 staff just dedicated to this and a massive effort that's absolutely on an exponential trajectory. So our vision with the center is to provide world-class head and neck cancer care through innovation, persistence, and game-changing research. And this kind of gives you a sense of what we're envisioning. For those of you who are wondering how I put this together, I didn't. My wife works at Enterprise. She told me that my slides weren't very good and that I need to make better slides. So this is a byproduct of our discussion. So this is the key slide, she tells me, and I do agree with that. The idea is that we want to not only create equitable access to care, but we want to put the patient at the center of the care. And from there, we can talk about new comprehensive treatments, profiling tumors, and then combining those things to actually perform integrated research that then feeds back on new ways of preventing and treating this horrible disease. So we're really, for this effort, putting the patient at the center. We now have a navigator that will actually be the patient's cheerleader. Many in this room, if you ever have had any type of medical issue, probably benefited from having a family member there at their side through thick and thin. Many patients aren't so fortunate. And so the idea is, how do we give every person a cheerleader, someone they can ask questions to that'll make sure they know exactly where they need to be, get appointments together, make a very challenging time as seamless as possible, coordinate the care with all these different people that surround the patient in their orbit throughout their treatment. But just improving care is not enough. The reality is, as much as I love to tell you that we're using the robot, the reality is about half of our surgeries involve huge operations that take 12 or 14 hours. And so we're fortunate to have the resources to handle those. But the question is, how do we do better? How do we literally put ourselves out of business in a way that we don't need to do invasive surgeries or keep patients in the hospital even one day? How do we make cancer a chronic disease? That can only happen by actually understanding what's happening at the level of the biology. And so as part of this tumor center effort, and with support from everyone in this room as well as similar efforts, we're hoping to establish a pipeline for actually sequencing thousands of these tumors, put them through artificial intelligence algorithms, and then begin to understand what's actually happening at the individual level of a tumor. So I want to give you a sense. It's fun to talk about things in generalities, but I'm sure you all want to actually hear about something real, something we're really working on. And so I want to spend just the last few minutes showing you what we're doing in the lab, and I hope it piques your interest. So just to start with an analogy, up until now, most sequencing of tumors, you take a piece of tumor you might get from the operating room or the clinic, you grind it up, and you get the DNA or the RNA out of it, the building blocks that tell the cell what to do, how to make and continue propagating the cancer. But the problem with this is, just like taking a fruit salad bowl and grinding it up to make a smoothie, you get some sense of what's going on. Maybe there's some strawberry, maybe there's some pineapple. But there's probably plenty of stuff in there that you wouldn't even notice if it was added in. So over the past decade or so, there's been real interest in how do we actually look at the pieces going into the smoothie? How do we understand each little piece of fruit that contributes to the fruit salad, or in this case, to the tumor? And so what we're able to do is actually sequence each individual cell by taking a piece of the tumor, barcoding that cell, and then we know exactly where the DNA or RNA came from. So for the first time ever, we can say, look at a tumor and say, of this tumor, there are 10,000 cells. There's some that are rapidly proliferating, not surprising, we're talking about cancer. There's some that are responsible for the cancer spreading. There's some that are hiding out in the corner saying, don't look at me, I'm not a bad guy, but is responsible for the recurrence. And so When we did this across patients, it was really interesting. You could see each patient here colored in a different color, and we use a computer to actually cluster the cells. So each dot here, each single dot is one cell from one patient. And at the 20,000-foot view, if you look at this, you might have the same feeling I do, which is it's very disheartening, because what this shows is that every patient's tumor is incredibly different from every other patient. But it turns out when you actually zoom in on each patient, you start to see the same sort of programs come out, programs related to the cell cycle, spread, et cetera. And one of those programs, as you can see in the lower left, which we call partial EMT, is responsible for the cancer invading and spreading. And this is something we found in head and neck cancer. What's really amazing on the right is you can see that patients with this program, as shown in green, have much worse survival at 5 years than patients who don't have the program, shown in red. And I think what's actually even more amazing than that is if you take this program, you ask the simple question, does a patient have this partial EMT program? Were they a smoker? What stage were they? What was their gender? What was their race? This program predicts their survival better than any of those things. So if you walk up to head and neck surgery, you say, what's the one thing you'd want to know about a patient besides their stage? I think you'd uniformly get the answer, what's their smoking history? Now I can tell you I'd rather know this thing that came out of our data, this genomic marker, than even that piece of information, which shows you the power of what we're trying to learn and do here. We've now expanded these efforts with the support of WashU as well as Barnes to over 192 patient samples. The data I showed you a minute ago was from about 7 years ago. And in that short time, we've gone from looking at 2,000 cells, which I just showed you, to 1.5 million individual cells from 190 different patients who were kind enough to provide their tumors. And what you could see is this massive set of different programs that come out of these tumors as shown in the right. All the different pieces of fruit contribute to these different flavors in the cancer. And by understanding this, we can start to really understand what's contributing, what's driving the cancer forward. How do we attack each of these different programs in different ways? Once you remove a tumor after surgery, you may not care to target the program that's causing the cancer to grow. The cancer is gone. You want to target the cells that are hiding out in the corner. And so this gives us that first insight into what are those cells, what do they look like, how do we begin to identify them, how do we begin to target them. And interestingly, we've now expanded this across many, many cancers. So in collaboration with Itai Tarosh at the Weizmann Institute in Israel, we've actually been able to look at almost 3,000 tumors from across the body and look at over 5 million different individual cells. And I could talk about this for hours, but one thing I'm excited to tell you is this partial EMT program, which is labeled as number 4 here, it says mesenchymal, is actually found in many, many different cancers. So what we're learning is that what we're finding in head and neck is actually probably relevant across the board. And maybe that doesn't seem surprising, maybe it does. But I think it's exciting because it tells us that what we're learning might go beyond just our narrow view of head and neck cancer to all of oncology. And the other thing we're able to do is actually take these data, make them accessible to researchers across the world. So this data from 5 million cells is available to anyone who can log in, look at it, of course, in an unbiased way. There's no patient information involved, and they can start to analyze this data in a hugely accessible way. So to Tim's point, we're taking something that we've done here and magnified its impact 100, 1,000, 10,000-fold through our work. The other thing we're excited about is if we can understand what's going on at each individual cell level, we can start to compare patients who receive treatment. So these are samples on the left. This is about 50,000 cells from patients treated with immunotherapy, which many of you know is a very exciting form of treatment, highly effective in lung and melanoma. But in head and neck cancer, it only works 15% of the time. So what we've done is compare patients who get immunotherapy, get a biopsy of their tumor before treatment and after treatment, and ask, how do we predict which patients are going to respond? How do we augment what's happening to make the drug work better? And this is really where I think the future will go. So in the last minute, I just want to show you how we're pushing the boundaries a little bit further than even what I showed you. So to extend my analogy, we started with the fruit smoothie. You grind up the tumor, you look at what's in it. I showed you the single cell data where we look at each piece of fruit that went into the smoothie to understand what it was, how many there were, all those different flavors. But what if we could actually know where that piece of fruit came from? We could actually know where it was in the tumor. So now we're not just understanding what type of cells are there. We know exactly spatially where they are in the tumor. Add an entire dimension of data. And so you could see here a slide. This is a representative patient tumor that we've done something called multiplexed immunofluorescence. And on the right, you can see all the cells we've analyzed from different tumors. You can see in tumor number 26, we looked at 827,000 individual cells from this patient. And for each of those, we know exactly where it came from on the slide, in addition to knowing what type of programs were present. And so looking at this, you can see a slide here. We can break it up into its individual components. That's the nuclear marker. This is an epithelial marker. This is a marker called LAMC2, which marks partial EMT, vimentin, potoplanin, P63, which marks head and neck cancer. We can take this, we can segment out the tumor and the stroma using machine learning. We can apply different segmentation approaches, then do this across the whole slide. So we're looking at a massive amount of data on each individual patient, which is really the key. And then asking questions like, for a given cell that's responsible for invasion, what are its neighbors? And how are the neighbors supporting this cell to allow it to invade, to allow it to spread? Ultimately, our goal is a molecular tumor board. So if we have all this sequencing data, if we have all this genomic data, we can start to ask the tough questions that have been impossible to answer. Why do 5 patients with the exact same cancer do well compared to 5 patients with the exact same cancer who recur within 6 months? That's really the question that we need to be answering. And without this data, without having it readily available at this depth and this knowledge, it's absolutely impossible to answer that question. So again, thank you so much for your attention. Thank you so much for being here. I really hope you feel energized by the efforts that you're involved in. We couldn't be more grateful, and it really, really is an exciting time for all of oncology. So thank you so much.
Perfect job. So you can see the power of these teams we put together and the acceleration and the pace of discovery. So Sid, thank you. So our next speaker is Dr. Patricia Ribeiro, and she is going to talk about, she's actually a radiologist, but she's actually looking at gastric cancer. And it turns out that gastric cancers are sort of interesting because they frequently hide antigens. In other words, they put them intracellular, and many of our therapies don't work very well. And so what she's actually doing is trying to figure out, gee, how in the world do we increase the expression of these antigens so that we can better treat the cancers? And by the way, she has some really impressive pictures. Patricia.
Thank you so much for the introduction. I wanted to start off by really thanking you for believing in our research and for really being partners with us in what we're doing in the lab, and I will show you in the next few slides some of the things that we're working on. So one of the tumor types that we're working is gastric cancer, and we really need to detect cancers, not only gastric cancers, but other tumor types. We really need to detect them as early as possible. And here are just some cartoons on the left. If those tumors are detected at an earliest stage, around 7 in 10 people will still survive after 5 years. Now, if it's at later stages, less than 1 in 10 people will survive. So we really need approaches that enables us to diagnose at as early as possible. And one of those techniques is positron emission tomography. And here's some of the images going from early stages to late stages. This technology has really potential because it enables us to visualize tumors in the whole body and in real time. And so each one of these dots represents disease. And so if we look at the late stage, the image on the right, you can see all of these different lesions. They're all representing the disease. And in the lab, we believe that positron emission tomography is powerful because when we think about tumors, they're very heterogeneous. They are different from one person to another, as we can see in this cartoon on the left. In the same tumor type, the cells will group in these different clusters, and all of these clusters have different levels of heterogeneity very different from one another. And even at the single cell level, tumors are very heterogeneous. So we really need the approaches such as positron emission tomography that enables us having a full picture of these tumors. And this is important as I'm trying to show here. So comparing a primary tumor with a metastasis. So the primary tumor in the left here could be classified as low expressor or negative for a certain antigen or receptor for which we have antibody therapeutics. And so in this primary tumor, if this antigen or receptor is not there, this therapeutic won't bind to this tumor. Now, when we compare primary and metastasis, they are very different. And so if in the case of this example here, this metastasis is positive for this antigen, and so now these therapeutics will bind to the metastasis, but we won't bind to the primary tumor. And so if we think about collecting these different lesions through biopsies, it's impossible to collect all of the different lesions in one person. So in PET, we can have access to that information because we can have those images in the whole body and also in real time. And so the image on the right is actually from a woman with breast cancer. This patient, the primary tumor was classified as negative for this antigen, so this person won't be eligible for this antibody therapy. On PET imaging, which is the image on the right, it was detected metastasis, which expressed this protein. And so because of this expression, this person was enrolled into a therapeutic, an antibody therapeutic that could recognize those. And in this person responded to the therapy as seen in the image at the bottom on the right. And so in my lab, we pair the potential of positron emission tomography PET imaging with these biomolecules, which are called antibodies. And so the images at the bottom are just images from transmission electron microscopy showing how these molecules look. And think about these molecules as molecular spies, which when injected, they will travel through the body and they will accumulate at these different tumor lesions. These biomolecules are very, very specific for proteins or antigens in these tumors. And so here's some images of some of the preclinical data. When these biomolecules, these antibodies, accumulate at the tumor site, they will bind their receptor and they will accumulate at the tumor. And so then we get PET images such as these ones. And the tumor on the right is just an extracted tumor from these models. So the colors in here from blue to red means more uptake for this biomolecule. And you can see how this drug, this antibody, distributes in different ways throughout the tumor. So some of the things that we're working on is how can we increase this, the density of these antigens, these receptors on the surface, so now when the drug gets to the tumor, we have more accumulation and we have more drug delivery to these tumors. And so we're exploring some biological approaches towards that, and as you can see here, we can highly increase the signal. Now we have more of this red signal, and we have these hot spots, meaning that we have more therapy going to these tumors. Another thing that we are working on is tumors are very heterogeneous, as I was saying. They're smart. So when we uptake or when we target one of these antigens, one of these receptors, they will find ways to upregulate others. And so they will continue to grow over time. And in the lab, we're working on approaches where we can target multiple things in the tumor at the same time. And we're working on this technology that we call antibody click, which received funding from the NIH for 5 years with the potential to get 2 more extra years, so a total of 7 years, where we're working on this technology targeting multiple receptors on the tumors. The images on the right at the bottom look like, it's how these molecules look like. They interact with each other. And what they also do is when that click happens between them, it results in an enhanced internalization, more drug delivery to these tumors. From an imaging perspective, this is really important because when we think about, again, between primary tumors and metastasis, And being that they are different from each other, with the conventional approaches that we have here, so let's say the tumor on the right expresses this antigen, but the tumor on the left does not. So with the conventional approaches that we have, these antibody molecules will not accumulate on the tumor on the left. So you can see how this technology uptakes different tumors that have different levels of these different receptors. And so with that, I really would like to thank again for supporting our research, and it's a pleasure to be here tonight. Thank you. This just kept moving. I don't know why.
Dr. Perez, thank you very much, and I promised that you would get some really great pictures. All the radiologists, they always have these fabulous pictures in 3D, and so pretty amazing. Our next speaker is Sheila Stewart, and Sheila is one of our more senior participants at Siteman. She is the Associate Director for Basic Science for the entire center. But Sheila studies the environment of tumors, how one cell may influence another cell to let a cancer grow. And she's now in the process of getting a breast spore grant, and we have 3 in leukemia, in endometrium, and pancreas, and we're aspiring in breast and head and neck and brain and a few others that are in the process of being submitted. So, Sheila?
So I wanted to also add my welcome and thank you. I think Tim just called me old, but I will forgive him for that. So I wanted to spend a little time, more experience. See, I like him. Anyways, I want to spend a little time really telling you how my group has leveraged the Frontier Fund and how you've made a huge impact on our ability to do foundational discovery, which is still the bedrock of where our next therapies are coming. If we're ever going to get to that, personalized medicine, we have to understand the underlying fundamentals that drive cancer. And so we have been interested for a very long time in just this question. Why is there really almost no cancer until the 5th decade of life? Okay? This is actually across the human population. Of course, there's pediatric cancer, but the bulk of cancer happens in the 5th decade of life and after. And you have to take a step back when you see something like this and say, why? What is it about getting old that influences this rapid increase? Now, many people, and Sid gave us a very nice introduction, there are genetic changes, mutations. We know these drive cancer. We know they're critical for taking a normal cell to a tumor cell. But we also know we have robust crews inside every one of our cells that spend their entire lifetimes scaling through the DNA and fixing all those problems. But they slip through sometimes. And the question is, how do those mutations slipping through drive cancer? Well, we know it's not one that slips through. We know it's a lot of them that slip through. We don't know the exact number, but we know it's a lot that takes that nice formed normal cell and turns it into that super ugly cancer cell that eventually goes on and kills people. And so the question is that the only thing that describes this? Tim already alluded to that I care about the environment. One of my standing jokes is I don't care about tumor cells, except Sid keeps telling me they matter. Now, of course, that's a joke. I care very much about tumor cells. But the question is it only mutations, or are there other things in play? We know mutations accumulate in age. This is real data from people. This is skin, okay? But we also know there are things called senescent cells. We call them old cells. They also accumulate with age. And we also know from work from our group and many others that they might be important. Now, we like to think of tumor cells as a big ball of tumor cells, but as actually both Patricia and Sid have already alluded to, that's not what they are. They're incredibly complex. They have a lot of different cells. Sure, they have tumor cells. Of course they do. It's cancer. But they've got a lot of non-tumor cells. And those non-tumor cells are important participants in the ability of that cell that's getting those mutations to become a cancer. And in fact, we now know that when you mix them together with indolent or very poor and wimpy cells, they can take that wimpy cell into an aggressive cancer cell. Think of it this way. You got a really good kid, starts hanging out with a lot of druggies, and he really becomes a jerk, all right? Your environment can have a huge influence in your outcome. And so we got really interested in how this happens. And many years ago, we actually discovered that those old cells, they make everything yummy. You can imagine if you want to be a tumor cell. They make things that help tumor cells grow faster. They make things that tell the immune system, back it off. We do not want you to attack this happy little cancer cell. So they do a lot of different things that help drive that tumor cell or that tumor cell to become a tumor. I was lucky enough to get a Frontier Fund award. They funded something the NIH refused to do because it might not have worked. Sid already alluded to this. And we ended up discovering that a specific protein, all you need to know is it's P38 today. Who cares what it does, except it helps you make that yummy stuff. And if we could block it with lots of different scientific ways, evil genetics or drugs, we could actually get rid of all that yummy stuff. And so we wanted to ask, well, if we could do this in a bunch of plastic, what happens if we're looking at real tumors? And so all you need to look at this is color. More color is more cancer. Less color is less cancer. And what we could show with these little mice that have a lot of metastases is if we give them P38 inhibitors, we stop it, we block those normal cells from making all that yummy stuff, we could reduce tumor metastasis to the same extent of paclitaxel, PTX, which is still used in the third line of therapy in metastasis. So as a single agent. Now, yes, they're mice, but these are the preliminary types of data that we need to start to have to base clinical trials on. Again, this would never have happened if it hadn't been for the Frontier money. Now, when we started looking at it, I think Sid might have shown it. You might have noticed we kept looking over. That's because we could see the slides in the window, not we didn't want to see you guys. But I think he showed you a Kaplan-Meier curve. This shows us how long you survive. And I hope what you can appreciate is if you don't get any treatment, and you're a mouse in this case, you're the red line. That's how long you survive. This is a very aggressive tumor. But if we put you on paclitaxel, chemotherapy, or on the P38, we actually get a little bit more survival. But the question is, it's a little bit more. Okay, it's as good as chemo, but it's not better. How do we make it better? I'm not going to tell you about the 10 years of work that took to get here. I'm going to tell you that we figured out who the target was. We figured out why P38, when we stopped it, was actually reducing the tumors. And it also gave us a little insight into how we could make this better. A picture is worth 1,000 words. We were able to leverage the P38 inhibitor plus an immunotherapy. It's called OX40. You don't care how it works today, just know it's an immunotherapy. There's no tumors left in those mice. 100% of those animals are cured of metastatic disease. Now, can this happen in a person? There's only one way we're going to figure that out. But we would never have gotten here, again, without the Frontier Fund. Last part before I go. It's not just about helping our patients live longer. It's how do we help them live better with the time that we afford them. Unfortunately, a lot of our therapies come at a cost. They induce different types of toxicities. And can we start to think about how we reduce this? Well, we got into this because I love graduate students. One day my student came running into my lab. She's like, Sheila, you're not going to believe this. The mice are a lot happier. And I'm like, back that off a little. Can you define a happiness? We're talking about mice here. She's like, well, they're just running around. Much better than the mice that are in chemotherapy. Remember, they lived just as long, right? So we started thinking about this, and we realized that most of our mice ended up passing away because they had huge metastasis to the bones. 70% of our breast cancer patients that are metastatic have lesions within their bone. This can cause great problems for them. Pain, fractures, hyperglycemia. Now, we do have some treatments for this to reduce the symptoms. It doesn't slow down the metastasis, but it helps them with their symptoms. And we just knew because we have an amazing bone group at Washington University, and this really underscores the interactions we have with many people that necessarily don't work on cancer. We knew that that P38 thing that makes all that yummy stuff also is really important for bones. So we went looking at the bones. And this is a CT scan through the femur of our mice. All you have to notice of PTX, paclitaxel, there's a big hole. That's not good. Inside our bone, we should have a lot of stuff that helps the stability of it, our bones and the mouse bone. Remember, the PTX treated and the P38, they all live the same amount, but the bones are in much better shape. Between the better outcome and the fact that we had the equal reduction in metastasis, starting hopefully this summer, we're waiting for final FDA approval. We will be launching a clinical trial looking at the effect of our patients on these drugs. Can we give them a slightly better quality of life, and can we slow down their metastasis? We're going to find out. We would not find out if it wasn't for you guys and those who were here before you. Finally, this is what we think about when we think about chemotherapy. This is what's on all the tabloids, right? This is what's on the news. But we wish this was the only toxicity our patients suffered. There's actually a whole slew of things that can happen. Luckily, they don't all happen. But when we started thinking about what was happening in our animals when we reduced this P38, for a lot of reasons, when we get back at the bar, I'd be happy to explain how we got here, but we had reason to think that neuropathy, this is the stinging pain or the numbness that many patients on chemotherapy receive, have after their chemo. Luckily, it resolves in many of them, but not all of them. We had reason to think that these same pathways we were tracing might affect that. So this is actually looking at the version of the mouse. Think of it as your fingertips. And all you have to know today is that those ugly little red lines going up are the nerves of the mouse going up into their fingertips. I know they don't have fingertips, but please go with the analogy, okay? Importantly, when we get chemotherapy, all the little lines are gone. They've lost their nerves, okay? That's not good, because when that happens, we can actually poke the mouse and see if it can feel. If it takes a long time to feel us poking them, don't worry, it's not sharp, it's blunt. We know they're numb, and if they pull their paws back quickly, we know they're in pain. And so basically, when you see an increase on this chart, that means they're numb. So this is exactly what happens to many patients. They're on chemo, they become numb. Could we actually reverse that by attacking that P38 or those old cells? This is a little bit complex, but mice on chemo, you see how they increase and they're numb? And unfortunately, just like many of our patients, if you let them go longer, it can compress to pain. Now it's actually taking, when I touch them, they're pulling their hands back very fast because they're in pain. And what we could show is if we got rid of those particular cells, that numbness took them totally back to normal, arguing that perhaps this approach might also be able to effectively treat neuropathy. Again, these are mice. But these preclinical data are the type of data we need if we're going to move forward and potentially help our patients live better and longer. And with that, I just want to use one last minute as an analogy. I know this is probably overused, the iceberg, but the Frontier Fund, I think that Tim already said, venture capital, it gives us the versatility to look underneath the surface. We all know that underneath the surface is what took the Titanic out, right? It's all of that stuff underneath. Those are the mechanisms that create the cancer that's on top of the water. I'll give you a perfect example. That is where immunotherapy came from. And if you are lucky enough to be those patients with melanoma or lung cancer that respond to it, this is a cure. And this came from 20, 25, 30 years of those dorky scientists in their little white coats, hiding in their labs, just trying to figure out what was underneath the surface. And now we are making huge impacts on those patients. That's my underneath the iceberg. All of this was based on a grant that I got a decade ago, so now I admit it twice, from the Frontier Fund. And so that led to a clinical trial that we all hope will make a huge impact in our patients. And with that, I do need to end with the most important, which is the people that actually make this happen. This is my group. They're amazing. And thank all of you very much for everything you do.
All right, so we're going to have a quiz. Nobody's like, we've locked all the doors. Everybody doesn't get out of here until we ask a really good question. Okay. Are there any questions? John?
Question is about the process of these approvals for NIH funding. So it starts with maybe a grant from the foundation, and good research gets done, and then it turns into an NIH proposal and a grant, and the leverage is incredible. But my question is, it just seems like a lot of the approvals are very experienced researchers, and maybe a lot of the younger researchers have great new ideas, and do they come to the forefront, or they have to wait their turn, kind of thing? Because these younger people might have totally different, bold ideas. That's all.
Do you want me to take that? That's a great question. Keeping the pipeline alive is so important. So first I'll give you my own anecdote, and then I'll say what we're trying to do. So I've tried to point out, over a decade ago, I was one of those young people, thank you. But those bright ideas would not have happened. The NIH would never have funded it. So we are doing that. Importantly, the money that goes to the Siteman investment program, over 50% of that is going to our young people, and the overall return on investment is 8-to-1, which means those investigators are turning over those funds and getting those grants. We absolutely have to support those bright ideas, those things none of us have thought about yet. And I think we're doing that very effectively in the Siteman investment program.
So I'll just add, of our senior leadership in the Cancer Center, of which Sheila is a part of that, 3/4 were homegrown. So they all, like Sheila, started out as an assistant professor and worked their way up through the ranks, as it were, because of the mentoring and because of the opportunities that the Siteman Investment Program provided, et cetera. And so we actually have a pretty good track record for doing that across our cancer center. So other questions? Yes, sir.
It was alluded to, but it wasn't addressed directly, the use of AI in what you guys are doing today. Can you speak to how AI is accelerating the whole process, and how excited are you about the potential impact of it?
Yeah, I'm happy to start there. I think one of the most obvious places we're using AI more and more is image analysis. And so the ability to kind of handle these multiplex, multi-dimensional data, you really need artificial intelligence to parse through it, because there's literally so many combinations that you could look at that to pull out the signal from the noise is very challenging. A version of that I alluded to is when we sequence all these tumors, it's great to have sequencing data on 500 tumors, but as Sheila said, they have many, many mutations. So how do you figure out what's important? So that's another example. How do you pull that out and then begin to infer what you predict based on AI and see how it matches? So we can actually take data sets we train on, right? We train the algorithm, we train the computer, and then validate them in a separate data set to see if the computer actually is doing its job good enough, how good is it doing, and is it actually an incremental or even substantial improvement over, say, the prior iteration. And through this sort of process that we can repeat over and over again, we can actually make tremendous strides in thinking through the data. The first step, though, is to have the data, and I think we're finally there, that we're generating these massive forms of data, and now the analytics are catching up with what we have.
Oh, there's another question in the back.
Yes. I wanted to know if your studies include a study of H. pylori, and if there is also a relationship between stomach and head and neck cancer directly or indirectly.
You want to talk about the gastric?
Yeah.
Yeah, so I would say in terms of stomach and head and neck cancer, there are some common risk factors between patients, but beyond that, we don't know of a strong kind of etiologic, meaning what causes the cancer link. We do know that patients with head and neck cancer are much more likely to have lung cancer and esophageal cancer, as well as many other cancers. So sometimes, for example, with head and neck cancer, we'll start the procedure by actually checking the esophagus for cancer first, because it's so common that we don't want to miss that, and then treat the head and neck cancer only to find down the line that the patient has a very large second tumor. We're not aware of a link between H. pylori and head and neck cancer, but in terms of the relationship to gastric cancer, defer to my colleagues if anyone has any comment.
It is a link between H. pylori and gastric cancer with certain priorities, but again, H. pylori, at least that I know of, is not linked to head and neck cancer or other types of cancer. Many of the other genetic abnormalities are, and I have to say, one of the aspects of Siteman that is really unique is people not only form these teams, but the teams actually work together, talk to each other, present to each other day in, day out. And so things we learn in breast cancer, we can oftentimes apply to head and neck cancer, or things we learn in head and neck cancer, we apply to gastric cancer, etc. And we have many, many examples where it would have taken us decades on our own to figure out the sequence, the right therapy. But we were able to do that because somebody said, gee, you know what? This treatment of NK cells works really well in lymphoma. And so one of our medical oncologists, who by the way at the time was pretty junior, said, well, what about in pediatric cancers, leukemias, where those children that get recurrence have a very high risk of dying? Could we use NK cells? And as you'd imagine, there was a lot of work behind the scenes. In the first 17 patients treated, 15 of them had complete responses. So if it hadn't been for this interaction at a meeting in the institution, we would have never figured that out. Other questions?
We're going to take one more question, and then our physician researchers will be available for the post-reception for questions as well.
I'm curious, there are a number of, shall I say, inferior cancer centers in the rest of the country and internationally. But that's because I'm biased toward us. But how much sharing goes on between what we're doing at Siteman and other cancer centers, both nationally and internationally, and how does that work?
Yeah, there's actually extraordinary cross-fertilization between centers. As you would sort of imagine, you know, Sid or Patricia, one of these physicians who are treating a particular cancer, know who the experts are around the country. And so if we don't have a clinical trial that may fit a particular patient, we can pick up the phone and call somebody, etc. In addition, much of our work now is given freely. Just like Sid was saying, you know, this whole database, although it was created here, worked on here, invested in here, is available to anybody in the world scientifically who wants to access the database. And fortunately, we protect all the patients whose tumors create the database, but it's available. And for example, we probably have more genomic information at Siteman Cancer Center than any other center in the country. And all of that has to be given freely to the rest of the scientific community. So we're pretty used to that and have these little groups between various cancer centers, et cetera.
Thank you so much, Dr. Eberlein, and to our wonderful panelists. We'd now like to welcome Jim Meyer to the stage for closing remarks.
Thank you, Tim, and thank you to you all. I mean, sharing the stage this evening after you all have presented is truly humbling. You are the ones that are doing the work, and I want to thank you all for your promising research. The work that's being done at Siteman is truly amazing, and I am honored to be a part of the illumination as an ambassador and as a board member with our terrific leaders, Carolyn, Julie, and Rusty. So I want to ask everybody here a question. If your doctor told you tomorrow you had cancer, are these the kind of people you want on your team? I see a lot of people nodding their heads. And I'm gonna answer for you. The answer is yes. I was diagnosed 10 years ago with a rare form of pancreatic cancer, and since that time, I have been receiving treatment from people like this and the other extraordinary physicians at Siteman. And so, as you've learned tonight, our support of the Cancer Frontier Fund allows Siteman physician scientists to pursue the research that's going to lead to better treatments and healthier lives for everyone in our community. So, today we're asking you to do this. We're asking you to commit to the brilliant Siteman physician scientists that are working diligently to find cures, and to commit to showering hope on the thousands of patients that are fighting cancer. The way that you can change lives is as simple as sponsoring a table at the Illumination Gala. It's as simple as raising your paddle at the Illumination Gala during the Fund a Cure to make a heartfelt gift that inspires everyone. When you give to the Cancer Frontier Fund, you're part of this incredible progress that we heard about tonight, and that will save more of our families and our friends. I hope you will join me by getting involved in the illumination this year. That's what we're here for. We cannot afford to stand on the sidelines. So please mark your calendars for June the 1st. At the Ritz-Carlton right here, and every sponsorship at the illumination, every paddle raised during the Fund a Cure, and every auction donation, every gift at the gala, it all moves vital cancer research forward. You might have heard, celebrity host is comedian and TV late night icon Jay Leno. So this is going to be a fun night, and it's going to be an inspiring night, and it's going to be a night to remember among friends coming together to do great things. Please visit the Foundation's website for more details about the event, and look for your invitation in the mail if you have not already received it. So now we simply invite you to continue the conversation and talk with our incredible physician scientists and other guests tonight as you enjoy cocktails and some hors d'oeuvres in our reception area. And I thank you again for joining us, and I hope you all have a wonderful night. That's all.